Hey everyone, this is Sanskriti Varshanay from Molecular Detectives. Here, I share information about cancer, from the molecular level to things in our daily lives and environment that affect cancer development. My goal is to explain the intriguing science behind cancer in meaningful episodes that will help you understand preventative measures, cancer science, and treatment methods, and ultimately for you to make evidence-based decisions to minimize the risk of cancer. Thank you for joining me in today's episode, and I hope you enjoy. Cancer is a word no one wants to deal with. It happens when cells replicate uncontrollably and even spread to other parts of the body and destroy healthy organs. In this episode, I'm going to talk about how genes work, what mutations are, how inherited and acquired genes play a part in cancer, and finally, some specific genes that play a role in cancer when mutated. Let's get started. Genes are pieces of DNA that have instructions to make proteins that carry out specific tasks. Genes are compiled into chromosomes, which are in your cells, and there are hundreds to thousands of genes in a chromosome. Genes are made up of nucleotides, like how a sentence is made up of letters. Nucleotides are building blocks of DNA and RNA and consist of three parts: a sugar, which is deoxyribose in DNA, a phosphate group, and a nitrogenous base. There are four nitrogenous bases: adenine, cytosine, thymine, and guanine, or A, C, T, and G. So there are four types of nucleotides, and each gene has a long sequence of nucleotides, and the order of the nucleotides determine the specific code that genes follow to make a specific protein. Now, let me tell you about the central dogma of biology. This is a theory that states that genetic information flows from DNA to RNA to a protein. Let me explain. In order to make a protein that carries out a specific function, specific instructions are needed to make it. Those specific instructions are called genes. So the first part of the central dogma of biology is genetic information flowing from DNA to RNA. In this step, a cell selects a gene, which is a specific part of DNA, and an enzyme called RNA polymerase attaches to the start of the gene. The RNA polymerase uses the DNA strand as a template to build a strand of mRNA flowing from DNA to RNA. In this step, a cell selects a gene, which is a specific part of DNA, and an enzyme called RNA polymerase attaches to the start of the gene. The RNA polymerase uses the DNA strand as a template to build a strand of messenger RNA, or mRNA, by matching DNA bases to RNA bases. This process is called transcription and happens in the cell's nucleus. The next process is genetic information flowing from RNA to a protein. This process is called translation, and it takes place on ribosomes in the cytoplasm. Here, mRNA goes to our ribosome with the genetic message and it binds to it. And remember that the mRNA is made up of many nucleotides. So the ribosome reads codons, starting with the start codon, and tRNA, which is transfer RNA, transfers amino acids that correspond to the codon to the ribosome. A codon is a sequence of 3 nucleotides, and eventually the ribosome reads the stop codon, and the resulting sequence of amino acids is finished, and it makes up a protein. So the central dogma of biology is really lengthy, but it's important because it shows exactly how DNA is read and then turned into proteins. Understanding this process will make it a lot easier to understand how mutations can change proteins into damaged ones and eventually lead to cancer. Now let's talk about mutations. Mutations are when nucleotides in a gene change. For example, instead of a DNA sequence being AGGTC, the mutation changes it to ACGTC. This specific mutation is called a point mutation, which is when a base is either added, removed, or switched in a DNA sequence. The change can seem minor, but specific codons code specific amino acids, so even one change would produce a different type of amino acid. Sometimes a mutation may change a codon in the middle of an mRNA sequence to a stop codon. This means that the signal to stop adding amino acids to the chain will happen earlier than it's supposed to, and the resulting protein will be too small. Or a mutation may cause a stop codon to actually change to a codon that codes for some amino acid. Since there's no stop sign, the protein would keep growing and growing. And I just gave examples of mutations in the translation process, but honestly, they could affect processes anywhere with DNA. Now, how do these mistakes cause cancer? Well, some errors in genes that lead to altered versions of those genes or gene variants may cause big mistakes, and some may cause smaller mistakes. Some gene variants may result in a protein that functions the same as the original protein was supposed to. The error in the gene is basically not important because it didn't change anything. Some gene variants may result in a slightly different protein that might be like a little bigger, for example, so it doesn't function as well as the original protein should. And some gene variants may result in a super different protein that doesn't work at all, or it has a completely different function than the original protein was supposed to. Since it changed the protein completely, the mutation that happened in that gene would be really bad because now there's a protein in your body that's not working in the way that it should. And that mutation's effects could be really worse if that specific protein that got changed was really important or if it worked with a lot of cells in your body. So the placement of the mutation on the sequence can affect the severity of the mistake and also what it does to the body. That protein change could make it harder for your body to break down toxins from tobacco smoke, according to the American Cancer Society, or it could even make you overweight, which is a risk factor for cancer. The body usually has measures to help combat these mutations, but when the mutations pile up, then severe changes can happen and lead to cancer. Now, I want to clarify, not all mutations are bad. Some can also be good. Like the reason why people have different colored hair and eyes is because of mutations in the DNA sequences that code for your hair and eyes. So, mutations make us different from each other, but some can lead to cancer. I just explained a lot, but it's all important to help you understand how mutations lead to cancer. Next, we'll discuss inherited and acquired gene mutations and their involvement in cancer. Inherited gene mutations are passed down from parent to the offspring. Since this mutation comes from a parent, it's present in the gamete or germ cell. And once this cell reproduces to form other cells in your body, the mutation is present in every single cell. This mutation isn't the sole reason for causing cancer, but since you have a mutation in every cell, it makes it more easier to have more mutations more often, so you're more likely to develop cells with more mutations that can result in cancer cells forming. You're also more likely to develop cancer earlier in life than people who don't have inherited gene mutations, since you already have one from birth. This type of mutation is also called a hereditary mutation or germline mutation since it gets passed down in families. Some examples of hereditary genes with mutations that may be passed down are the BRCA1 and BRCA2 genes, and mutations with these genes are associated with breast and ovarian cancer. That's why it's so common for people whose family members have had breast or ovarian cancer to worry about getting the same type of cancer. Your parents have two of each gene, and if one of them is mutated, then you have a 50-50% chance of getting it or not getting the mutated gene. So even if someone's mother had breast cancer, they may not get it because they got the normal gene from their mother. The other gene mutation is an acquired gene mutation. This type of mutation happens when a person develops it in their life. For example, the mutations that I was talking about before, when a mutation in a gene causes a protein to become altered from the original protein, that's an example of an acquired gene mutation because that happened during your lifetime and it wasn't there before. These mutations can happen randomly or because of environmental causes like constantly spending lots of time in the sun, exposed to UV radiation, or being exposed to chemicals in your workplace environment. The good thing is that you can't pass on these mutations to your children because they don't develop in your egg or sperm cell. This mutation is also called a somatic mutation or sporadic mutation, meaning that it happens in your body cells, not your sex cells, and it happens without warning. Now, I want to dig a little deeper into specific genes like TP53, BRCA1, and RAS and how they cause cancer. First, let's start with TP53. The TP53 gene is known as the guardian of the genome. This gene has instructions to make the p53 tumor protein. TP53 is a tumor suppressor gene, and a tumor suppressor gene is a gene that regulates and slows down cell growth and division. TP53 helps the body get rid of mutations to prevent them from harming the body and potentially turning cells into cancer cells. For example, if UV radiation damages DNA, then TP53 determines if that DNA can be fixed or not. If the DNA can be fixed, then TP53 gives instructions to other genes to fix the DNA. But if the DNA can't be fixed, then TP53 instructs the cell to go through apoptosis, which is programmed cell death. The main goal is for the cells with a mutation or DNA damage to not divide, or else more cells will also have the DNA damage or mutations. TP53 also makes sure that cells are dividing correctly and that they don't divide uncontrollably. If they do, then TP53 triggers apoptosis in those cells so that they don't spread. But what happens when TP53 itself gets mutations? That's when things start to go downward. The body has genes that work to protect itself from harmful mutations, so even like a few mutations in your body are fine, because other genes take care of making sure that they don't cause harm to other cells. But when genes like TP53 get mutated themselves, they aren't able to protect the body from those mutations. Mutations in the TP53 gene cause the p53 protein that results from it to be different than normal, so it's not able to function properly. Without the p53 protein to make sure that cells are dividing at an appropriate pace, cells may divide too fast or too often. And because the p53 protein isn't regulating the cells, tumors are more likely to form. If the cells that are dividing too rapidly have DNA damage, then all new cells that result from those cells will also have DNA damage, which will hurt the body. Additionally, the mutant p53 protein can increase cell division instead of just not functioning, which leads to more cell growth. Mutant p53 can also make cells resistant to chemotherapy and radiation therapy treatments, and it can promote cancer cells to spread to other parts of the body. And this is a process that is called metastasis. So, not only can mutant p53 protein not do its job, but it can also have very negative effects on normal cells and cancer cells. The TP53 gene can be inherited or acquired. An inherited TP53 mutation can cause the Li-Fraumeni syndrome, which is a syndrome that makes it more likely to develop cancer such as breast cancer, osteosarcoma, which is bone cancer, brain tumors, and even leukemia. An acquired TP53 can also cause these cancers and even liver and lung cancers. Now, let's move on to another gene called the BRCA1 gene. While BRCA1 doesn't have a special name like TP53 did, it's also an important gene, and the protein it makes is called BRCA1. This protein helps make sure that cells are not dividing too fast and uncontrollably. BRCA1 also repairs damaged DNA, and it works with other genes to fix breaks in DNA that may be caused naturally or from chemicals in the environment and other risk factors. Mutations in the BRCA1 gene cause the BRCA1 protein to not function as it should, which means that it's not effective at stopping cells from dividing too often or uncontrollably. This can lead to a lot of mutations happening or damage to DNA, and those cells with the mutations and damage will keep reproducing, which can lead to developing cancer cells and tumors. People who have BRCA1 mutations have an increased risk for developing breast cancer and ovarian cancer, especially in women. According to the National Institute of Health, women with a BRCA1 mutation have a 50 to 80% risk of developing breast cancer by age 70. Men with BRCA1 mutations are also at risk for breast cancer, pancreatic cancer, and liver cancer. Finally, let's talk about RAS. RAS is actually a gene family, and these group of genes are also controlling cell replication and apoptosis. They stimulate cell growth and influence cell migration. However, mutations to these genes code for RAS proteins that are permanently activated, which leads to uncontrolled cell growth and eventually the development of cancer. Mutations in the RAS gene family are usually acquired and are actually one of the most common types of mutations. They can cause pancreatic cancer, colorectal cancer, and lung cancer. That was an information-packed episode. I hope you now have a better understanding of how genes work, how mutations can cause cancer, inherited and acquired gene mutations in cancer, and specific genes that can cause cancer when mutated. That's it for this time. Thank you for listening, and stay tuned for the next episode.